Hi guys, welcome back to my channel. Today we are going to be doing a quiz on biochemistry of nucleic acids, and there are 70 questions. I'm Dr. HF and I want you to see how prepared you are. So are you ready? Let's go. I want you to think about the answer before it comes up. So get your pen, your paper, and start. Which researchers demonstrated that DNA contained genetic information in 1944? Avery, McLeod, and Mackay. What is the biological term for a DNA segment containing information for synthesizing a protein or an RNA product? Gene. Good. What are the three characteristic components of a nucleotide? A nitrogenous base, pentose sugar, and a phosphate group. A molecule consisting of a nitrogenous base, pentose sugar, without a phosphate group is called a nucleoside. To which carbon atom of the pentose sugar is the phosphate group esterified in a standard nucleotide? 5' carbon. At which specific nitrogen atom do purine bases covalently bond to the pentose sugar? N9. At which specific nitrogen atom do pyrimidine bases covalently bond to the pentose sugar? N1. Moving on. Name the two major purine bases found in both DNA and RNA. You should know this. Adenine and guanine. Which pyrimidine base is unique to DNA, rarely occurring in RNA? Thymine. Which pyrimidine base is unique to RNA, rarely occurring in DNA? Uracil. Great job, guys. What is the full name of the ribonucleotide abbreviated as AMP? Adenosine 5 monophosphate or adenylate. What is the full name of the deoxyribonucleotide abbreviated as DGMP? Deoxyguanosine 5 monophosphate or deoxyguanylate. Which biologically important purine serves as the end product of purine degradation in humans? Uric acid. Name two purine bases that serve as intermediates in the synthesis of other purines. Hypoxanthine and xanthine. Which plant-derived purine base is found in tea? This is for the tea lovers. Theophylline. Which nucleotide analog is used clinically to treat hyperuricemia and gout? Allopurinol. Which sugar-modified synthetic nucleotide analog is commonly known as AZT and used to treat HIV? Zidovudine. What drug is used to suppress immunological rejection during organ transplantation by degrading into 6-mercaptopurine? Azathioprine. State Chagall's rule regarding the molar equivalence of bases in DNA. Chagall's rule. Number of adenine is equal to thymine and guanine residues equals to cytosine. Why do single-stranded DNA and most RNA not obey Chagall's rule? They lack the complementary base pairing of a double-stranded DNA. Who proposed the double helical structure of DNA in 1953? James Watson and Francis Crick. The two strands of a DNA double helix are described as dash because they run in opposite directions. Antiparallel complementary. What is the standard width of DNA double helix in angstrom units? 20 Armstrong or 2 nanometer. In the Watson-Crick model, what is the length of one complete turn of the DNA helix? 34 Armstrong or 3.4 nanometer. How many nucleotide pairs are typically found in one complete turn of a bDNA helix? 10 pairs. The backbone of a DNA strand is composed of alternating dash and dash units. Deoxyribose phosphate. How many hydrogen bonds form between adenine thymine base pair? 2. How many hydrogen bonds form between a guanine cytosine base pair? 3. Well done, guys. Let's keep going. Why is a GC base pair approximately 50% stronger than an AT base pair? Because it contains 3 hydrogen bonds while AT has 2. In a double helix, the genetic information resides on which strand? Template or sense strand. Which DNA conformation is a left-handed helix? Z-DNA. Which DNA conformation is observed under conditions of relative humidity and presence of sodium or potassium ions? A-DNA. How does the major groove of A-DNA differ from that of B-DNA? A-DNA is narrow and deep, while B-DNA is wide and quite deep. What type of non-Watson-Crick hydrogen bond allows for the formation of a triple-stranded DNA? Hookston hydrogen bonds. What novel tetrameric structure is formed by polynucleotides with very high guanine content? G quartets or G tetraplex. The phenomenon where DNA loses its helical structure and the strands separate is called denaturation. Does DNA denaturation break the covalent phosphodiester bonds of the backbone? No, it only breaks the hydrogen bonds between the strands. What wavelength of light is used in a spectrophotometer to measure the absorbance during DNA denaturation? 260 nanometer. What is melting temperature of DNA? Temperature at which half of the helical structure of DNA is lost. How does GC content affect the melting temperature of DNA? The higher the GC content, the higher the melting temperature. In eukaryotic cells, DNA associates with basic proteins called dash to form chromatin. Histones. We're almost halfway. What is the basic unit of chromatin? Consists of DNA wrapped twice around the histone core. Nucleosome. Which four types of histone proteins make up the nucleosome core? H2A, H2B, H3, and H4. Why is DNA resistant to alkali hydrolysis while RNA is susceptible? DNA lacks the 2-hydroxyl group on its pentose sugar. Which histological color reaction is used to identify RNA due to the presence of ribose? Arsenal color reaction. Which nucleotide serves as the principal form of chemical energy for cellular metabolism? ATP, adenosine triphosphate. Give two examples of nucleotides that function as second messengers for hormonal actions. Cyclic AMP and CGMP. Which uracil derivative is involved in the synthesis of glycogen? UDP glucose from UTP. Which class of RNA transfers genetic information from genes to ribosomes for protein synthesis? Messenger RNA. What percentage of total cellular RNA is typically comprised of ribosomal RNA? 50 to 80%. What specific structure caps the 5' terminal end of eukaryotic mRNA to prevent hydrolysis? 7-methylguanosine triphosphate. What is the function of the poly A tail found at the 3' terminal end of mRNA? To provide stability and protect from exonucleases. The sequence found at the 3' end of the acceptor arm of all tRNA molecules. CCA. Which arm of the tRNA molecule contains the 3 specific bases that recognize the mRNA codon? The anticodon arm. Why is the D arm of tRNA so named? Because of the presence of the modified dihydropyridine base. Eukaryotic ribosomes are composed of which two major subunits? 60S and 40S subunits. Which 3 types of RNA are found in the eukaryotic 60S subunit? 28S, 5S, and 5.8S. What term describes RNA molecules that possess catalytic activity, such as ribonuclease P? Ribozymes. Well done, well done. In plant purines, caffeine is derived from coffee, while dash is derived from cacao. Theobromine. What is the primary role of transfer RNA in protein biosynthesis? Acts as an adapter molecule to translate mRNA information to a specific amino acid. Which type of RNA is involved in the selection of proteins for export from the cell? Small cytoplasmic RNA. Which histone core component is not part of the nucleosome bead? Histone H1. What provides the structural framework for ribosomes? Ribosomal RNA. How long is the DNA molecule found in a human cell when fully extended? Approximately 2 meters. The distance between two adjacent nucleotide pairs in a bDNA helix is approximately 3.4 Armstrong or 0.34 nanometer. Which specific RNA is mostly present in bacteria and adds peptide tags to incorrectly synthesized proteins? Transfer messenger RNA. Which purine derivative is involved in transmethylation processes? S-adenosyl methionine. If there is 10% thymine in a DNA strand, what is the composition of cytosine present in percentage? 40%. What is the function of a small nuclear RNA? It's involved in mRNA processing. What is the sequence of the complementary strand shown here? That's the sequence. We've come to the end. That was a fun quiz. Let me know what score you got in the comment section and study hard. Thank you.